Yes, good afternoon, class. I am welcoming you this afternoon to Metric Hub, our YouTube channel that is simplified for every learner. Actually, education is simplified through Metric Hub. So just know that you are connected to Metric Hub. Uh, Subscribe and also turn on your notification button to keep posted of our uh, revision, our lessons, and every other information that is going to be posted on the channel. Thank you so much. Without wasting any time, we are just going to introduce and we are going to start on our first module of medical practice N5. Just note that our module is divided into two papers. I'm sure that you know that medical practice is divided into two papers, paper 1 and paper 2. These two sets of papers together, they make the learner's final mark of exam, where actually in your book it is labeled as part A, and which is paper 1, and there is also part B, which is paper 2. The presentation that is here is actually comprehensively articulated and covered, starting with paper 1. All definitions of terms and relevant examples are given, as well as in paper 2. At the end of every chapter or every section, I'm going to give you a series of past typical questions or question papers that are also going to be accompanied with their memos, which you are going to use for your evaluation and also just for practicing to get ready and become familiarized with your examinations, techniques that you will see within medical practice. These questions are not in PowerPoint anyway, but they will be consistently posted and presented as Word documents, which you will see attempted at every other stages that are to come. So thank you so much for joining us and thank you for joining Metric Hub and all things as well. Without wasting time, let's go to the next topic or next subject. We are going to start on chapter 1, chemical pathology, which is actually paper 1. Like I said, is section A, which is paper 1, lesson 1. There's some definition of terms that you must understand here. Chemical pathology. Define chemical pathology. It is the study of chemical and biochemical mechanisms of the body in relation to diseases, mostly through analysis of body fluids, such as blood and urine. So in terms of this, you should understand that chemical pathology, it actually looks at biochemical mechanisms of the body in relation to diseases, and also through the analysis of fluids such as blood and urine. They are the fluids that are analyzed, where in many cases or in many diseases, these are significant changes. There are significant changes in chemical composition of fluids, such as the raised blood enzymes. I'm sure that you remember to say enzymes. What are they? What are enzymes? Due to their release of from the heart muscles after heart attack or raised blood sugar in diabetes mellitus due to lack of insulin. So what I'm just presenting to you is that we do have some changes in chemical composition of the body, which is caused because of infection or diseases, which can be either heart attack or it can be also the problem in balancing of some hormones in the body. I'm sure that you remember what is insulin. Insulin, it is basically a hormone. It is a hormone, this one, a hormone that is responsible for converting glucose to glycogen. If you remember in this regard, in our science subject, remember that insulin is actually helps to avoid chances and problems of diabetes mellitus, which is called sugar in other terms, which is a condition that is caused as a result of excess of glucose in the body, which is actually as a result of either inadequate insulin or absence of insulin in the body, which would result in high sugar levels, which they call diabetes mellitus. So please just note that for the interest of others and learning, we can just have to understand that there is a relationship that is there, which students must also know that glucose is converted into glycogen, and then what happens there is that there is a reverse relationship which takes place here, which is supposed to be understood by many learners, or actually by almost every learner must understand that that is how it is, the relationship of glucose and glycogen, and also what happens there. So what we are saying is that what increases here, it is insulin. It is insulin, and then insulin, it actually decreases the levels of glucose in the body. And then underneath here, we have got glucagon, which we are saying it actually helps to what? To decrease. It decreases. I mean, actually, what happens is that, sorry, in this regard, glucagon, it actually helps to convert glycogen into glucose, which is one other thing that you must understand, that glucagon, glucagon actually it increases levels of glucose in the body. It actually increases the levels of glucose in the body, this one, the glucagon. So what I'm saying is that glucagon increases, it increases glucose level. It increases glucose level. So what I'm basically saying is that please note that this is just a reverse relationship that is supposed to be understood by any other science student who is into or who is pursuing this medical field of study in regard to how does insulin cause diabetes mellitus. So that is basically a simplified diagram that can be used in explanation to in regard to that. So tests are designed to detect these changes qualitatively and quantitatively, and then they are compared to results from healthy people. So what we are saying is that there are tests which can be done with regard to the analysis of body fluids that we are referring to, such as blood and urine. And these tests are quantitatively or qualitatively checked or evaluated, which is compared, or when they are compared to results of healthy people. So that is what we mean by chemical pathology. And then chemical pathology, it also includes the general or routine chemistry, which is commonly ordered blood chemistries, e.g. the electrolytes, blood gases, the lipids, liver and kidney function tests. I am sure that you must also understand that it actually it also includes the general routine chemistries, which are just to say the composition of the electrolytes, the blood gases, the lipids, and also the functioning of kidneys. Special chemistry elaborate techniques such as electrolysis and also the manual testing methods. That's what we mean by special chemistry. And then I would want us to look at the next one, the pathology. Pathologist definition. Define pathologist. Pathologist is a physician who interprets. The pathologist actually interprets, he diagnoses the changes caused by disease in tissues and body fluids. So in regard to that, he is a physician who practices or evaluates or supervises diagnostic tests using materials removed from living or dead patients, and functions as laboratory consultant to clinicians, also to who conduct experiments or other investigation to determine the causes or nature of disease changes. So in other words, what you need to understand is that a pathologist actually traces the changes and the building up of a disease in a human body, understanding in regard to the balancing of the body fluids and other any other tissues of the body, which we say that these are done using materials that are removed from living or dead patients, in regard to that, a pathologist, he works as a consultant to clinicians and also conduct experiments and other investigations that would help to determine the causes or nature of disease changes that a person is suffering from. And then let us quickly look into the pathologist's duties and responsibilities. A pathologist's duties and responsibilities: one, analyze, we are saying he analyze and test samples using microscopes and other testing tools. These include samples of blood, urine, tissue, and other bodily specimens. So in other words, you need to understand that a pathologist has the responsibility of testing samples, which he can test through microscopes and other testing tools, which we are saying these samples can be of blood, they can be of urine, and any other bodily tissues, which we say they are bodily specimens. He ensures accuracy of test results, that the tests are done are all correct. He ensures accurate. We are saying the correctness, that tests are correct. We are saying correct ensures that the tests are correct, which is one key point that we need to understand, that accuracy or the correct, that ensures that the results are accurate or they are correct. And then write reports. He writes also reports, perform results analysis, and hence he spent most of the time working in the laboratory. One thing that you should understand is that as you can see, it has to do with the chemicals and chemical composition or balancing of bodily fluids. So in regard to that, a pathologist spent most of his time in the laboratory, which we are to understand that he really works with patients, but reports in his research to a primary physician. And then what we are saying is that the investigations and the researches that he does are communicated to his primary physician, where we need to understand that he works uh, these pathologists work in specializations such as immunopathology, which we are saying that he can also understand the weakening of the immune system, what is causing the immune system to be weakened in regard to that. And then he also looks on chemical pathology, also understanding the deviations in chemical composition of the body, what are the causes of those variations, as well as for forensic pathology, and then molecular pathology as well. These functions are as a result of the fact that a normal human body is a balanced unit. One thing that you need to understand is that in healthy and in any in this discipline, you need to understand that the human body is actually a balanced unit, which when a disease occurs, the balance is disturbed, as well as the chemical composition and fluids. So a pathologist would identify these imbalances that might arise in the body, and then identify and pick up to say that what are the causes of those imbalances in terms of the chemical compositions and the fluids, and then understand again that the body has got various units, but it works as one. Thus, the extent of a disease is diagnosed eventually through samples collected in each case. So in terms of treatment and curing of patients, the pathologist actually helps in treatment of diseases. Is actually the center that provides the background towards the treating and handling of diseases that individuals or patients are suffering from. So please understand that the pathologist is actually the backbone towards diagnosis or towards the treatment of a disease, by understanding the chemical pathology or understanding how this disease came into being, and also the possible ways that it can spread and how it can be cured. And then we need to look at the duties and the duties of medical secretaries when dispatching medical samples. These are just basically other areas of interest which medical practice students must understand. We are saying the duties of medical secretaries when dispatching medical samples. Remember that we said in our first chapter there that a pathologist works with medical samples. So these samples are received or they are pegged and assembled by the medical secretaries. So it is always the duty, it is always the duty of the medical secretary that the samples are sent correctly and following the specific applying rules and regulations within or towards each of the samples that are to be sent. One, we are saying the medical secretary ensure that a correct order is used for a specific sample. So in regard to this, what is supposed to be understood is that we have a variety of of orders that can be used, which we are saying that we have got e.g. For example, we have got fluids orders, we have got fleshy. And then what we are saying is that these are not placed, or they are not held in what? In they are not placed in the same type of water. Each type of sample has got its specific water. Hence, it is the duty of the medical secretary to ensure that a corrective sample holder is used. And then ensure samples have correct amounts in holders. That is the quantities. That is the quantities must be correct in the orders there. The quantities must be correct. The medical secretary must ensure that there is enough or there is correct quantities or correct sample amounts that are within the orders that are to be dispatched for medical examination. And then ensure sample orders must have best minimum information. Best minimum information must be on the what? On sample orders. Best minimum information, as you can see, we are saying the doctor's name, patient's name is also supposed to be on the sample order, the dates on which the samples has been collected, the test required, and also insertion of a separate form for the person's address and medical fund number, which can also be, for example, in regards to the paying of the bills, the medical fund number. So what we are saying is that it is the duty of the medical secretary to ensure that the minimum information that is required on the sample holder is placed there. So we can basically sum up by saying that the medical secretary ensures that information that is on the sample holder is correct and corresponding, e.g. the name of the patient, the doctor's name, the dates and the test to be done, as well as a separate form for addresses and other medical fund numbers are inserted. Next one, we are saying sterilization of containers to carry samples for bacteriological examination. So we are saying these are samples that are analyzed to identify possibilities of bacteria. We are saying these are samples to be analyzed to determine chances of what? To determine chances of bacteria in them, to determine possibilities of bacteria. Hence, we are saying it is the duty of the medical secretary to ensure that they are sterilized. Sterilization helps to kill the bacteriological, what you call contamination that can be within the, or which can be on the containers or in the containers. Hence, these are supposed to be well sterilized to avoid the possibilities of contamination, which would give unreliable information or give biased results with regard to the samples that would be put in there. So the medical secretary again ensure urgent arrival of samples and provide preservative measures for in case of any delays that might arise. So we are saying timeless delivery of what? Of samples, it is duty of the medical secretary. In regard to this has to do with timeless deliveries of samples and safekeeping of them. Safekeeping is also very important. The medical secretary, we are saying he has to ensure that the samples are delivered according to the times that they are supposed to arrive to their centers of examination, or else they he or she can as well provide some preservative measures that would ensure that if ever there are delays for in terms of the transport movement, the samples cannot turn bad. So we are saying that he has to ensure that there is preservation facilities for samples in order for them to be fresh and usable for the purposes that they are intended for. And then ensure stock replenishment consistently to avoid shortages. This has to do with the ordering. The medical secretary must consistently order. The medical secretary, he must consistently, in regard to this, we are referring to the point of the medical secretary must consistently order, he must consistently, must consistently order stock, order stock on a timeless basis. So the medical sector actually must order stock to avoid shortages or running out of stock. So it is the duty of the medical secretary to ensure that all the equipment and all the things that are supposed to be used during the processes or examinations and every other procedures to be done, the relevant resources are in place. So must consistently order stock on timeless basis to avoid shortages. It is quite important that the medical secretary to avoid shortages. It is the duty of the medical secretary to do that. So please note that it is quite important that you note some of these responsibilities, as to say they are the key duties of the medical secretary when we are dealing with the key responsibilities of which that's the trade that you are in. And then types of samples. There are many different types of samples that are received for microbiological, this for microbiological testing in various laboratories. These samples fall into three major groups, and then specimen, which we are saying are generally taken for biochemical examination. These samples or specimens are taken for biochemical examinations. So in this case, we are saying samples for pathology testing include fluids. We can say they include fluids and secretions and what biopsies are also supposed to be there. So what we are saying is that the samples itself is usually small and is regarded as being a representative. It is actually a representative of the material from which it was collected. For instance, we can talk about a 10 milliliter specimen of blood. A 10 milliliter specimen of blood, representative of the blood from the body, where it was taken from. So in regard to that, what we are saying is that a sample is usually, or it is actually supposed to be just a representative or a reflection of the whole of where it was collected from. So with reference to that, please note that it is actually a representation of the entire body or of the entire fluids from where the sample was collected from. So you've got urine sample is usually taken from the surgery, or the patient takes it from home and bring it for examination, as examples of samples that we can talk about. I'm sure that you know, for example, when we talk about pregnant women or pregnant mothers, they often get urine samples or urine tests. In terms of that, the urine is taken from it can be taken from the surgery, or the patient can take it from home and bring it for medical examination or chemical analysis. In regard to that, we also need to understand that a clean wide mouth bottle is used. The urine must be taken early in the morning. That is one key thing. The urine must be taken early in the morning and must be midstream. It must be midstream urine. Midstream, what we mean by midstream, we are just referring to say it mustn't be the first urine that comes out when a person is urinating. It must be found midstream sample. It must be found in the midst of the urine, as to say there is a first that comes out, and then when the person is urinating, can let the first urine come out and then take the in between or the urine that is coming before the process of urinating is done. So what is happening, it is actually called midstream urine. It is called midstream urine. Because why is it supposed to be that? It is actually clean and actually provide the best uniform and the best information that is required with regard to that. So the patient must be put at ease to avoid nervousness in order to have the urine come out. I'm saying in cases where the urine is collected from surgeries, the patient must be put at ease to avoid nervousness. So sometimes you would find out that uh, in terms of our examination, they will ask you to say, why should a patient, or give the major guidelines or the major conditions that a patient must have when collecting urine samples. We are talking about calmness is the key factor that must be there. Avoid nervousness, keep the patient calm, and then the urine can be produced or can be released. And then this term here, maturation, please note, uh, mutilation, it is always asked, it is quite often asked that you are asked to define the term. It is an act of urination. The act of passing urine or the act of urination is mutilation. That's what I mean, it is one of the terms that you might sometimes also find when you go to examinations. We have got cerebrospinal fluid as another form of sample or a type of sample that can be collected. It is, it requires specialized equipment in terms of collection of cerebrospinal fluid. Remember that it can also be the cerebrospinal fluid. This is the fluid which is found in ventricles, which is found in the vertebral column in a human body, used which is actually used for examination of possible diseases and other conditions that a person can be checked. So what we are saying is that it is usually taken at hospitals because of the specialized equipment that is required there. So this includes a lumbar puncture where a needle is placed between the third and fourth vertebral column into the spinal subacromial space. The subacromial space is actually a space where we have the cerebrospinal fluid contained. If you can check, even from the human skull, we have got what is called the dura mater and the pia mater and so forth. There's what is called subacronoid. It is just a ventricle, a space that contains cerebrospinal fluid, which is supposed to move and lubricate the joints and body reflexes that the body must go through. So a patient is bent forward, so the vertebra will move apart. It will move apart a little bit, and the first sample is collected, is moved apart, and then the first sample is collected. So that's what we are saying is an example of other fluids that samples that we are talking about. And then another sample, we can talk about diapers, which are also used for bacteriological examination. Diapers are also used for bacteriological examination as to identify possible chances of contaminations that can be found from our patients. And then a dry diaper is of no use if not placed in a special transport holder. A dry diaper. So in terms of this, diapers must be kept what? They must be kept moist, or they must be kept wet in their original conditions. Where a special transport holder is, it must be used, so therefore it can reach the pathologist as soon as possible. These are taken from IC. We can talk about diapers taken from IC, from the nose, from the throat, as well as the vagina, and then rectum, that is the large intestine. The rectum, we are saying large intestines. So what we are saying is that they are actually taken there as as to understand, to say that diapers are taken in both in places which they are used to as a covering, in to avoid either there's the running of nose. We can talk about the running of eyes. We can talk about the clearance of throat, as well as also some vaginal secretions that can be released, which we are saying this one from the rectum. We can talk about the anus, where they can also be placed what you call diapers, which we are saying in cases where there is abscess, where an abscess is covered with a scab, the scab must be lifted, and then the diaper then taken of the pus that is underneath. It is actually done in order to make sure that the real information or the real bacteriological contamination that is to be examined is collected from there. So we are saying where there is some covering that are done, we need to understand that the scab must be removed, and then the diaper then taken of the pus that is underneath, or the pus that is actually coming from either the wound or whatsoever it is. And then we've got tissue specimen is normally taken during an autopsy to determine the possible cause of death, of death in most cases for infections or cancer again. So the tissue specimen, it is normally done in order to identify possible causes of person's death and also any other infections. And after it is taken, it is cut, processed, colored, and then microscopically examined. I'm sure that you're saying that to say to see the bacteria, or what you call microscopically examined through, or are saying checked uh, through some microscope to identify any contaminations that are very minute and microscopic that might be there. So please understand that these are examples of what, of tissue specimens that we need to understand. And then uh, uh, just to finish up on this one, we have got microscopic examination, which is done. It is actually done by simply pouring the urine. It is just an examination of the urine, which is done where the urine is. We're saying urine sample is poured into a test tube, and centrifuging it or spinning it down in a machine for few minutes. We are saying where it is the top liquid part supertant is discarded. The solid part left in the bottom of the test tube, which is urine sediment, is mixed with the remaining drop of urine in the test tube, and a drop is analyzed under microscope to identify the crystals of red and white blood cells, as well as mucus ones, which we are saying these are microscopic examinations that are done to identify some urine problems that can be found in the urine. So we are saying urine color, this may vary from colorless to amber as a result of medication, nutrients, and diseases conditions that can cause the urine color to vary. One, we have got colorless, which means excess dilution and may result in diabetes. We are saying one, there is colorless, which results in which means excess dilution and may result in what, in diabetes. We have got milk color of urine, which result from pus present, which result from pus that might be present, albumin or fats that can also be there. Just take note of that. And then we have got orange color, is a result from certain remedies that a person might have been taking or might have been drinking, which would also cause the coloring to be orange. And then we have got red, certain coloring substances like beetroot. We can also, as a result of beetroot or of bleeding due to bilharzia. So bilharzia also, which can also cause the red coloring of the milk, I mean of the urine, is also a possible cause in terms of the red coloring there. We also have greenish, which is also jaundice or poisoning through the phenol or methylene blue. And then we have got dirty blue or green, which follows the decay of urine, also occurring in diseases like typhus, cholera. Poisoning with methylene blue and gall in urine causes the green color also. The gall, I'm sure that you remember to say, from the liver, we've got the gallbladder and stuff. So what we are saying, I mean, the gall, which we are saying that it can as well be there to cause the green coloring there, which we are saying it is contained or it is coming from the gall there. So we say the dark brown is also another color, which is dark brown or black, is due to bleeding, which is caused because of bleeding. It can be due to bleeding of urine tubes or hemoglobin, black water fever, and other diseases that are causing destruction of red blood cells, causes the brown or black color, dark brown color. We also have dark brown or red yellow is due to concentration of gall again, which is also due to concentration of gall again. And then there's also what is called smoke urine, presence of blood or tube in urine through excessive epithelium cells. I'm sure that with this, we can understand and remember that these are some of the areas of interest which you can be asked during an examination. So please take note that we are saying this is a quite important subject or topic which you guys should understand that the urine color and the possible diseases that they might cause. I'm sure you're going to enjoy. Thank you very much for your time. And please don't forget to subscribe and turn on your notification button. Thank you very much and have a good day. Thank you very much.